SANDHAN, all Gujarat integrated classroom.
સેટેલાઇટ ના માધ્યમથી જોડતી કડી એટલે સંધાન.
Good afternoon, students. Today we meet in a new academic year 2012-13 and we continue the program of SANDHAN in microbiology. This program has been well accepted and has been found to be very beneficial to the students. Today we are going to discuss and talk about a very important topic which is spoken about in the media as well as in the students community of life sciences, and that is the word biotechnology. Basically, at present students are seeking admission to various courses in life sciences and one of the subject is biotechnology. So biotechnology as a subject, what does it offer to the students? What are its scopes? What are the challenges faced by the subject of biotechnology? That is what we are going to discuss today. So a student should understand that biotechnology as a subject is, as we will see later on, is an interdisciplinary subject and has a wide number of topics and the subjects to study. It is not a one subject because basic subjects are physics, chemistry, biological sciences, mathematics, etc. But this subject of biotechnology is a gift of this century. The subject is a new one. It is, to be very frank, it is not a subject in itself, rather it's a combination of various different subjects of biological science, that is botany, zoology, parasitology, medical microbiology, human physiology, anatomy, as well as it also includes the other subjects of chemistry, even physics and engineering. So it's a kind of a multidisciplinary subject, and therefore a student has to have the basic knowledge of all other subjects in order to get a proficiency and expertization in biotechnology. So first of all, before I hand over mic to the next speaker, I would like to introduce her. She is Professor Kiran Trivedi, myself Dr. Rakesh Patel. We are both from R.G. Shah Science College, Ahmedabad, and we will be discussing this topic of scope and challenges in biotechnology. Now, what is biotechnology? What are the various definitions? What does the biotechnology mean? That will be discussed by Professor Kiran Trivedi. Madam, please.
Thank you, sir. My dear students, today the topic that we have selected is really very interesting, and as the discussion you will come to know that how interesting it is to know something about the biotechnology. As the sir told you that biotechnology is a word you all are very much familiar, but when we want to have something in detail, we should understand that what is biotechnology. So I will first begin with the term, the meaning for biotechnology. Biotechnology, actually the definition for biotechnology has been given by various scientists, various person or the various people who are working in the field, and this definition is according to their own experiences, according to their own experimentations. So if I have to tell you in general, the biotechnology is basically the utilization of biological entities or the cell of higher organisms, may be living or dead, or their components, which like enzymes, which are being used in the process. As a result of that, we get some product or we get some service which can be used in the welfare of human beings. So as a result of this, you will understand that the biotechnology is nothing but it is simply the use of living cells, maybe of higher organism, maybe of lower organism, maybe living, maybe dead, or it can be the use of cell components. Naturally, some of the definition, as I have told you, given by different people. One of them is the definition given by Bullock. Bullock said that controlled and deliberate application of biological agents, living, dead cells or cell components is biotechnology in technically useful operations either of productive manufacture or service operation. Now the point comes, what is biotechnology? So actually, as we understand that biotechnology is not a very modern term, but long back in the year 1919, the Hungarian engineer named Karl AGIC was the first one to use this term, and he said that any product that has been produced from the raw material with the aid of living organism, we can name that as biotechnology. And according to him, biotechnology means biology plus technology. Technology, as you understand, it is the application of scientific knowledge, skill, material, labor and wealth for practical purposes. Again, if we go for one more definition, it says that any technology that is associated with the manipulation of biological system is biotechnology. But in biotechnology, mainly we make the application of scientific and engineering principles in the processing of materials by using the biological agents, so that we get, in turn we get very good service and very good product. Now initially the production, naturally we may have one question that the production technologies which are related to agriculture, horticulture or animal husbandry may utilize plants and animals. They also generate the useful products, but we cannot call them as, or they cannot be regarded as biotechnology because long recognized and well established disciplines. Now, so what should be called as a biotechnology? Exploitation of animals and plant cell cultures in vitro for product or service as an integral part of biotechnology. So this is how we had come to know about the definition of biotechnology. Now, as sir has already told you that biotechnology is not a single branch, but it is a multidisciplinary science. It combines many more subjects like microbiology, chemistry, biochemistry. On the screen you can see immunology, cell and tissue culture, physiology, downstream processing, biochemical engineering. I mean, so many branches are integrated, and then we have formed this branch or this field as biotechnology. Next, here also you can see the different circles. Each one is representing different field of biotechnology: agricultural biotechnology, healthcare biotechnology, industrial biotechnology, whatever the word you may use, but they are all integrated with one another. So same is the picture showing all different branches integrated. This is again a same thing shown in the form of a tree, the different branches of biotechnology. Okay. Now, on what principle actually the biotechnological operations they work, or what are the various branches which are being involved. As I have already said that so many branches are being involved in biotechnology. Let us see. When we begin with the operations, suppose if you want to improve some strain, we begin with this improvement of the strain. So improvement of the strain, the procedure will make the use of subjects like microbiology, cell biology, botany, zoology and genetics. Once you have improved the strain, then you have to go for the mass culture that represents the physiology. Then after collection of the mass, you have to continue with the optimization of the microbial responses. Now these microbial responses are processed, or they are associated with the process engineering. Now once you have seen the responses of microorganism or their products, then comes the process operation. Process operation means during which the whole of the yield or the product has to be produced. So that the process operation will involve the various branches like chemistry, biochemistry, chemical engineering and biochemical engineering. So this is how we see that the biotechnological operations are based on different disciplines. Now coming to the next point, stages of biotechnology. Now biotechnology word may be given in 1919, but actually from the very old time, that is in ancient time, the people in the search of various things, they were wandering here and there, and they wanted some shelter, they wanted some food, they wanted so many things. So with the passage of time, they have started growing, and they have started discovering many different things. So ancient biotechnology includes the search of food and use of shelter and domestication of different kinds of animals. Second stage is a conventional biotechnology. Conventional biotechnology means it involves the fermentation promoted for food production and the production of medicine. Third stage is a modern biotechnology. It is a really very interesting one because it involves the manipulation of genetic information in organism, that is what we call as genetic engineering. Now let us see something briefly about the ancient biotechnology. Actually this biotechnology began with the early civilization and developments. There were developments in agriculture and food production. Actually we don't have very firm records about these things, but through the carvings and the sketches and any other archaeological research, we would come to know about this ancient biotechnology. Now coming to the history of biotechnology, you will find that there are many more developments were being made in the year 6000 BC, that is before Christ. Yeast was used to make the beer by Sumerians and Babylonians. Then in the year 4000 BC, Egyptians, they used yeast for making the bread and wine. 2000 BC, Egyptians, Sumerians and Chinese, they developed the technique for fermentation, brewing and cheese making. 500 BC, Chinese, they used moldy soybean curds to treat boils, that is to treat some type of infection. Then Indian ancient literature refers to Sura and Madira, which are basically the fermented products. This is a beautiful picture which shows that in the ancient time how the people used to make the wine by using the wooden big large vessels. Okay, coming to the next stage of biotechnology, conventional biotechnology. But biotechnologists, they were using the living organism for manufacturing a variety of useful materials, as for example fermentation of grape juice to produce the wine, antibiotic production by Penicillium chrysogenum, microbial production of glycerol by yeast, acetone butanol production by Clostridium acidobutylicum, citric acid production by Aspergillus niger, and production of streptomycin by using Streptomyces graeseus. Mutations, recombination and strain selections are the different kinds of techniques which were exploited or which were used to increase the yield of the product. Now let us see something more about the modern biotechnology. As the name suggests, it is a modern biotechnology, so naturally the techniques which are being used over here are very hi-fi techniques, and absolutely a new product is being obtained by the organisms. Here we are able to engineer a new genetic potential. That means we can absolutely create a new organism, new cell with the new genetic capacity. Technique that has stimulated the progress of modern biotechnology, they includes different one, as for example recombinant DNA technology, gene cloning, PCR technique, plant and animal tissue, protoplast fusion, protein engineering, biosensors, new reactor designs, computer aided bioprocesses. So these are the different modern biotechnological techniques. Now naturally one of them which is very important in biotechnology is the gene cloning. I'll just give you the short or in brief something about the how the gene cloning is being done. The first step in the gene cloning is the isolation of the target gene from the DNA of an organism. Second step is the insertion of targeted gene into the vector. Vector can be a plasmid or it can be a cosmid. Third stage is the transformation of recombinant DNA in the vector. Fourth stage is the screening of recombinants and selection of transformed cells. Finally, expression of a cloned gene through controlled element is very important. All these steps are dramatically represented in this picture. You can see the picture how the from the bacteria targeted gene has been taken, it is inserted into the vector plasmid, then that plasmid is inserted into another bacterium. Bacterium is a recombinant bacteria, it is permitted to divide, multiply, and so we increases the number of gene, and hence we increases the amount of product, maybe protein or maybe other type of product. So this is the outline of gene cloning procedure. Now the next topic, the scope of biotechnology, will be discussed by Rakesh sir.
Thank you, ma'am. See, madam has already outlined the biotechnology, what is it, and the definition of biotechnology. As she has discussed, biotechnology involves lots of subjects and it is a multidisciplinary branch. Now, since it covers two important things, that is, through biotechnology we get certain goods or certain products, that is one thing. And the second important thing is biotechnology sometimes is not used for getting any products, but rather it is being used in service industries. For example, we use cells, it may be of a microorganism or a higher forms of them, but these cells are being used not for production of any product but for service of human beings. Say for example, treatment of wastewater and bioremediation. Basically it is the microorganisms and lower forms of life which carry out such activity. So don't ever think that it is always going to be some product and it is also that product is going to be marketed and we are going to get money. But sometimes biotechnology is used for a service, and service is rather, I would say, a very important point as far as human life is concerned. It makes our life easy. So now when we talk about the scope of biotechnology, we will see in what way where the biotechnology has been used so far, and in future where it will be used, what are the potential fields where biotechnology can invade and can give certain results which makes certain products or make our life comfortable. Now broadly we will be dividing the scope of biotechnology into 5 different headings. One is healthcare and medicine, second is industrial, third one is agricultural, fourth animal biotechnology, and fifth one is environmental biotechnology. Now let us talk about the first one, each of them in brief. The first being healthcare and medicine. So in what way the biotechnology has contributed to the healthcare and medicine? Well, it has helped in diagnosis of diseases, it has helped in forensic sciences, in production of pharmaceutical products, in pharmacogenomics, in human genome product is also a type of a bio or technological invasions, and assisted reproductive technology. If we go into details, then diagnosis of genetic disease, infectious disease, DNA fingerprinting, which leads to the identification of criminals, rapists, etc. or culprits, and even the settlement of paternity disputes is because of the analysis of the DNA, that is its base pairs, by a process or by a technique known as a DNA fingerprinting, which is advancements in a biotechnological field. Then certain pharmaceutical products, which includes antibiotics, certain human protein replacements, certain products used for the therapeutic purposes of the human diseases, and vaccines. Apart from this, monoclonal antibodies also is a part of a biotechnological product. Pharmacogenomics tests or it checks the effects of drug and the correlation of that drug, genetic correlation with that of an individual. What can be the future? The future can be certain tailor-made medicines which are associated with certain specific diseases. Then there are certain accurate methods of determining appropriate drug doses, improvement in drug discovery and drug delivery, and certain better vaccines. As we can see, in future, when a person, as it is observed here, in future, when a person falls sick, as it is shown in the diagram, in the picture, he goes to the doctor at the top. The doctor checks the symptoms and he will order for various tests to be done. Once the results of the tests are available, then doctor will determine what type of sickness it is, and based on the individual's genes, based on the individual character, the doctor will formulate a drug, will deliver a drug to that individual, and the person gets well. As a result, you can see that a personalized medicine will be the branch of future. Every individual will be given a drug according to the nature of a particular person. This is possible because of a project which is known as a Human Genome Project, which was started in 1990s by many countries came together across the world, and within few years they analyzed the human genome. The complete sequencing of the human genome was done, and by 2001, that is within 10 to 11 years, the first working draft was published. Within coming 2 years, that is by 2003, the sequencing was completed, and as a result of it, it was found that human genome contains around 3.2 billion base pairs, whose average size of a gene is around 3000 bases. These sizes will vary, but the largest known human genome is of dystrophin, which is at 2.4 million bases. However, the number of genes which were estimated were much lower than the previous estimate of 80,000 to 1.5 lakhs. That is, the total number of genes in a human being is around 25 to 30,000. It was also found that 99.9% of nucleotide bases are exactly the same in all people. Only there is a difference of 0.1% in an individual. And at present, we know the functions of only around 50% or more of the discovered genes. Now, what are the benefits of this Human Genome Project? It has led to improved diagnosis of the disease, then use of gene therapy, the pharmacogenomics, that is to design the custom drugs as I have discussed earlier, and to formulate the drug based on the molecular information which has been given to us by Human Genome Project. Gene therapy, that is the genetic manipulation in animals or human beings to treat diseases, is also at the doorstep. It can be done by two things, either by augmentation of the gene, that is by inserting a gene, or by inhibiting a gene, that is by using an antisense gene which inhibits the expression of a dominant gene. And these two approaches can be achieved by either somatic cell gene therapy or germ cell gene therapy. Now, these types of germ therapy can be either be done in vivo, ex vivo or in vivo. Ex vivo means using the own cells for culture and genetic corrections, and then returning back those cells to the patient. For example, using a sickle cell anemic person, genes of those cells of those persons, correcting them, and again sending them back to the patient. While in vivo is by using a vectors which will deliver a particular desired gene into the patient's body. Cystic fibrosis and hemophilia can be treated in future by these kinds of gene therapy. These are the limitations or obstacles we call it, that is high costs. We have limited delivery tools, limited knowledge of functions of various genes, and multi-gene disorders, and what will be the effect of environment. All these are the obstacles, or what we call it as a challenges in biotechnology. However, as the researches continue, these obstacles or these limitations or challenges will be overcome. This is a cartoon whereby a human being says, or he thinks that, my God, the human genome has been unrevealed. That is, the base sequence has already been known. So man has to think to change the password as we change the password of our computer or our datas. Similarly, the outcome of Human Genome Project is also various tests, genetic tests which are being done. That is either by direct examination of the DNA molecule for its mutated sequences, and this can be used up for carrier screening, conformational diagnosis, pre-symptomatic testing, sex determination, or screening of the newborn baby, and even the parental diagnostic screening. Biotechnology can also be used, or the scope is also there in assisted reproductive technology, which is done either by artificial insemination, embryo transfer, or in vitro fertilization or embryo cloning. This is a figure which shows the embryo cloning. Early embryo is obtained from the recipient or from a donor, it is of identical cells. Then these cells are separated. Each cell developed into identical embryo, and then each embryo is implanted into different surrogate mother. As you can see at the bottom, there are 4 cows or the animals, female animals, in which this embryo is transplanted. As a result, we get babies who are having an identical nature, that is, the identical clone of our offspring is born. This is how the embryo cloning can be done, which will help us in getting our pet animals in a large number. The second aspect is the commercial industrial products. Biotechnology has been used widely in production of enzymes, antibiotics, organic solvents, then microbial polysaccharides, microbial rubbers, and even in biotransformations. It has also been used for production of single cell protein. It is also used in food and beverages. These are the classical kinds of fermentative products whereby the organisms, specifically microorganisms, have been exploited in many ways to produce these products. The newer branch is agriculture biotechnology, whereby the plant tissue culture, that is in vitro cultivation of plants, seeds and various other parts of the plants. It may be organs, embryo, tissues, single cells and even protoplast. They are grown in a nutritive culture medium under aseptic conditions. What are those applications of these things? It is development of pathogen-free plants, synthesis of economically important plants. Then we can also develop hybrids and even the protoplast generation. Micropropagation is also an offshoot of agricultural biotechnology. As a result, what will be the final outcome? We have a transgenic crops development. These transgenic crops, they are resistant to biotic stress, insect resistance. Biotic stress means they are resistant to insects, they are resistant to various pathogens like bacterial, fungal and virus. They are also resistant to abiotic conditions, which includes several pesticides, herbicides, resistant towards environmental conditions, that is drought or floods, resistant to snow, that is they have antifreeze protein in them, and resistant to saline environments, whereby the plants can grow in zones where the soil is saline. The further discussions of the role of biotechnology in agriculture will be done by Madam Trivedi.
Thank you, Rakesh sir. My dear students, sir has already told you a lot about the use of biotechnology in different fields, and especially the very important achievement in this biotechnological field. We have seen that the human genome has been sequenced so well, which was not possible without the help of this new field, biotechnology. He has also discussed different fields where the biotechnology is found to be so useful. He has told you that how in agriculture we can make the use of biotechnology. Now some more points I would like to elaborate at this juncture. Actually, when we talk about the agriculture, and we know that our country is absolutely based on agriculture, we are agriculture based people. So for us the agriculture is very, very important. Now let us see how this biotechnology is helping us in the field of agriculture. Sir has already told you that by using the transgenic plants, it is very much possible to create a plant that is found to be resistant to attack or damage by a variety of insects, viruses, bacteria, or any other things. Even the plant can be made resistant towards the herbicides, towards the drought, towards a very low temperature like the freezing temperature or the snow that we see on plants. Under all such adverse conditions, the plant gets the potential to grow. This is one of the greatest achievement in the field of agriculture. Not only this, but we have also seen that by this biotechnology we can improve the quality of fruits. That is, we can extend the shell life of fruits by using biotechnology. Not only this, but we can give the longer life to flowers. And one more very interesting thing is that we can give the desired color to and shape to the flowers. Apart from this, the transgenic for male sterility and for terminator seeds. The transgenic can be very important. Transgenic plants, this is how they can also work as a bioreactors. Now here the point comes, what is the meaning of bioreactors? So normally in the fermentation you have studied so well that we are having a fermenters and bioreactors, but how can a plant be a bioreactor? So just imagine that the whole plant making a variety of components, making a variety of compounds like a large quantity of carbohydrate, lipids, proteins, enzymes, or many other things, naturally becomes a bioreactor. Now we, with the process or with the help of transgenic plants which we get by the use of biotechnology, we can create or we can produce the plants which are very rich in carbohydrates, and because they are very rich in carbohydrate, they can become a very good source of starch and fructans. Even the plants can become very rich in lipids, so that we can have the production of short chain, long chain fatty acids, and even we can produce a rare type of fatty acids. With the help of transgenic plants, we can have the proteins, and the proteins are very special type because they are the therapeutic proteins. That means they can be used in the treatment of several types of diseases. Then we can have a large number of industrial enzymes, lysosomal enzymes. Not only this, but we can also be able to produce some antibodies and edible vaccines. Edible vaccines means what? You can insert a gene that produces a vaccine in some fruit which can be taken or which can be eaten up by us, and this is how that fruit containing a vaccine goes inside our body, inside the host, and so host gets vaccinated. So this is how the use of edible vaccines in case of by using transgenic plants is possible. Apart from this, there are many bacteria. One which you are very much familiar is a rhizobium. This rhizobium, as you know, is well known for its capacity to fix the nitrogen. It can fix the nitrogen, and thereby it can increase the soil fertility. So this is how this soil fertility in rhizobia, they are having a gene for this. This gene from rhizobia can be isolated and can be inserted in any other soil bacteria which will be able to again fix the nitrogen. So this is how we can increase the soil fertility by transferring the nitrogen fixing gene to other soil bacteria. One more important aspect over here which I would like to discuss is biocontrol of phytopathogen. Phytopathogen means the pathogenic organism affecting the plants, damaging the plants. They can be controlled with the use of siderophores. They can be controlled by some antibiotics, by enzymes, and by biofertilizers which are present in the soil itself. And not only this, but one very interesting point over here is that we can control the pollution in the environment by using different kinds of microorganisms or their products as biosensors. Biosensors are very good analytical devices which can be used, and they are having immobilized biological material. BOD, trichosporin cutaneum is the one organism which can be used as a probe which can help to detect the organic pollution within 15 minutes. We can also have gas biosensors, in which we are using organism like thiobacillus. This thiobacillus organism can sense or can detect the sulfur dioxide pollutant in the environment. We do have the immunoassay biosensors. With the help of immunoelectrodes, we can also sense the immune responses. Next point will be discussed by Rakesh sir.
Thank you, ma'am. See, as it has been mentioned that biotechnology does play an important role in management of pollution. But in what ways? As it has been discussed, it can sense the pollution by using various sensors, by various electrodes, by using various biosensors. However, not only detection, but it also helps in reducing the pollution. For example, reducing atmospheric carbon dioxide by photosynthesis and biological calcification. Next is very popular one, sewage treatment by bacteria and by use of algae. Then eutrophication, that is the enrichment of lakes and other water bodies, and removal of phosphorus from them. And even the management of metal pollution by a process of bioaccumulation. Various kinds of microorganisms, various kinds of biological systems are able to accumulate in their bodies, in their cells, certain metal pollutants. Finally, even the use of immobilized cells have also been carried out in order to manage the pollution problems. Next one is bioremediation. Bioremediation is a hot topic which is being discussed day to day, and here we use the microorganisms to clear the environmental pollutants. Mainly, broader methods are biostimulation, that is by enhancing the microbial activity by adding certain nutrients. As a result, microorganisms will grow, they will be stimulated to grow and will reduce the pollution, will eat away the pollution. Bioaugmentation, that includes the use of certain genetic methods so as to increase the biodegradation, that is bioaugmentation. Here in bioaugmentation, certain genes may be manipulated, certain genes may be inserted so that they have the increased capacity to deal with that particular pollutant, toxic compound. Now, these type of bioremediation can be carried out either in situ or ex situ. The details I will not be able to discuss in a short time, but in situ techniques, they can have either intrinsic bioremediation or engineered in situ bioremediation. Well, apart from pollution, biomass and bioenergy are also the fields where biotechnology plays an important role. In bioenergy, the energy produced from biological system, biogas is the foremost one. This biogas is a mixture of methane, carbon dioxide, nitrogen and hydrogen sulfide. Since methane is present in a high concentration from 50 to 80%, it can be used as a source of fuel. Since ancient times in India, biogas has been used for the fuel purposes. Even the days are not far where we will be using biohydrogen as a source of fuel. Bioethanol, that is the production of ethanol by fermentation, and even the other petroleum plants, that is plants having certain volatile fatty acids, volatile compounds, can be used for the fuel. Fuel crises are deepening day by day. As you can see, every day the price of petroleum products are increasing rapidly, and therefore biotechnology can answer and solve this problem to a certain extent. Then we also have transgenic animals. Transgenic elements means the animal which is created by using certain improvised quality of two different animals. That leads to improvement in genetic traits. It also helps to understand the human diseases. It can serve as a model for understanding human disease. It can be a product for medical and pharmaceutical application. It can also have improvement of quality and quantity of the products. And finally, the transplantation of histocompatible organs into human beings can also be done by using transgenic animals. Transgenic animals can also serve as a disease models, and they also can be used as a transformers. Transformers are nothing else but they are the dairy animals having a engineered capacity to express a particular protein either in a milk or in their meat, so that we have that particular protein through that animals. Even transgenic animals can also be used as a xenotransplanters. They are engineered to allow the transplantation of histocompatible organs into human beings. Transgenic animals can also serve as a food source and even as a scientific models. Now, so far we have discussed the various scopes of biotechnology, and believe me, the market for biotechnology is developing very rapidly. The worldwide biotechnology market is having a quantum leap due to the development in agriculture, industrial and animal biotechnology. At present, the biotechnology industry is more than 50 to 60 billion, and it is growing rapidly, more than any other industry in the world, that is at around 17 to 20% annual growth. The biotechnological markets in India are also very encouraging and promising, because we have got lots of infrastructure with us. India is expected to emerge with 10% market share in the global industries by the year 2010 to 2012. The advantages with India are excellent networks of research laboratories. We have rich biodiversity. We have trained manpower and extensive clinical trial opportunities, as well as well-developed industrial base. Due to this, India can definitely contribute vast in the field of biotechnology. Okay, last but not the least, what are the challenges in biotechnology? What we should look for in future? What are the limitations or drawbacks which we have to overcome in order to move forward? So, what are the points which we should take care of? And these points that includes, first one, ethical, legal and social implications of biotechnology. Now see, as we have seen, biotechnology can work miracles. Lot things can be done with biotechnology. Even the gene manipulation, human genome project, and the peeping inside into the human genome has led to the cure of various diseases. But at the same time, there are certain fears that since the modern biotechnology deals with the genetic manipulation, it may lead to certain unknown consequences. Are we playing God, or are we perfecting the genes? The somatic and germline germ therapy and the money involved in biotechnology. Huge amount of research investment is going on. Huge amount of expectation is there from the biotechnology field. And last but not the least is the racism eugenics. These are certain ethical, legal and social implications which needs to be answered. Basically, ethics is nothing else but it is a moral philosophy of what is right, what is wrong, what is good and bad, what is justice, what is virtue. And this is what individual has to determine. There are lots of positive and negative effects. Because of all these advantages, there are certain negative effects. What are those negative effects? We have to depend more on the developed countries. That is, developed countries have to depend on developed countries. Less labor requirement may lead to unemployment. That might be, a fear is there might be reduction in natural biodiversity and the ecosystem disturbance. Then there are certain legal and financial complications, that is intellectual property rights. Then manipulation of life. Since we are playing God, we are manipulating the cells. We have been able to manipulate bacterial cells. We are able to produce the insulin through bacteria. We have never thought of these things before 50 years. So manipulating this life, playing God, and this may lead to certain man-made evolution. This also contributes to genetic pollution, that is undesirable and uncontrolled gene flow into the population. This genetic pollution, it was a term coined by Jeremy Rifkin. Mr. Rifkin, a scientist and author, wrote a book, The Biotech Century, where he describes the risk that might occur due to unintentional processes of genetic modified organisms in nature. So these are the questions, and at present we do not have answers to all these questions. No doubt we can answer these questions to a certain extent, but no definite answer is available with us. So what will be the perceived benefits and the risk of biotechnology in future? What will be the environmental impact of biotechnology? All these things are the challenges faced by the scientist in future. Even the intellectual property rights of the farmers, researchers, private companies and public institutes, are these adequately protected? These are the questions which needs to be answered. So these are the challenges which are to be faced in future by biotechnologists, and I sincerely hope that in future the biotechnology is going to be a key word, a key contributor, and the coming 50 years are going to be for biotechnology. So with this, we would like to end our discussion. At the end, last but not the least, I would like to acknowledge on my behalf and on behalf of Madam Kiran Trivedi, our acknowledgements are due to the Commissioner Higher Education, Government of Gujarat, the staff members of the BISEC studio, and certain PowerPoint presentations which were earlier presented by myself and Dr. Pal, Professor Pal, and the topic was entitled Biotechnology and its Applications. We are thankful indeed to all these people for our successful delivery of the lecture and discussion of the topic. Thank you very much.
Thank you.
Namaste.
SANDHAN, All Gujarat Integrated Classroom.
સેટેલાઇટના માધ્યમથી જોડતી કડી એટલે સંધાન.